Thank you very much. It's really great to be here to introduce Jose. I met him 25 years ago, and since we are friends and collaborators, and I'm following his remarkable career. So Jose was born in Algeria, then his parents came to France. He went to medical school in Paris, and his residency in Strasbourg, and then came to the US to work with David Albert and John Dowling. And this really initiated his interest in research. He went back to Strasbourg, where Serge Picot joined him in Strasbourg as well. And they started a small laboratory in the basement. And no one wanted to give him money, but Jose is never bothered by that. So he started to develop new ideas for therapies. And people noticed this, that there is an amazing medical doctor who's interested in research. And then he was offered to come to Paris to start a new institute. And that became a now very extremely well-known and wonderful, vibrant institute, the Vision Institute of Paris. And he was also a chair at Coinsvin and Rothschild. Then building up an amazing community in Paris, he decided he wants to build another institute. So came to the US, to Pittsburgh, where he again built a wonderful research community, vision research community in Pittsburgh. So who is Jose? So for me, there are a couple of important points about Jose. Foremost and most importantly, he's a medical doctor who really, really cares about patients. We talk a lot, and many times he calls me and he says, you know, there I had this patient and I don't know what it is and I really want to figure out. And he cannot sleep for a week until he calls the whole world or he figures out and reads to really, really help that patient. That truly is Jose, who really, really cares about a patient. And everything for him starts with a patient. But he also is a scientist who loves to innovate. So when he started his career in Strasbourg, he was very intrigued that when rods die, somehow the cones, while sometimes they don't express the mutant gene, they also die, and search for a solution. And with Thierry Leviarati, they identified the rod-derived cone viability factor that over 20 years or more, he brought it to clinical trials. And this really became his passion, to do some science together with people and bring it to clinical trials. So the next one, LHO and gene therapy, he then brought it to clinical trials. And then electric implants. I remember around 20 years ago, he was very excited about this new technology. And then he always goes and works 20 years, and now it's again in clinical trials. Then we worked together a lot on optogenetic therapy. And finally, he really interested in clinical outcomes. He always tells me what we are missing is really good outcomes, and we should build more. So that's also his passion. But Jose is not only a medical doctor, a scientist, but he's a true real estate developer who builds institutes, not just because of the architecture, but because of the community. For Jose, an institute is a community that can bring together scientists and clinicians to have new cures for patients. He built a Vision Institute of Paris. Once it was ready, there was a fire. So I heard about it, I called him, I thought he's completely devastated, and he was more calm than me. He told me, don't worry, we rebuild it. And here it is, there is a vibrant institute. Then he appeared in Pittsburgh, and I was 100% sure I just have to wait a year, and there will be a new Vision Institute in Pittsburgh. And sure enough, it is there. Indeed, if he comes to your town, you can expect that there will be a Vision Institute. He's a teacher and collaborator who really sparks collective thinking and innovation. He builds communities and really likes to work together with people. I know that there are many people in this room who work with Jose, and all of us appreciate his dedication and his incredible devotion. When Jose tries to call you at 2 a.m., you can be sure he has a new idea what to do in research. Finally, he's a person who is really shaped by art, music, and architecture. So Jose loves these. If you enter his car, you will listen to the most amazing Bach music. if you enter his buildings, you will see visual art, modern artists he collected to show their pieces, and he loves to talk about architecture. So Jose is a real recognized leader who develops new therapies for the visually impaired. I listed some of his prizes on the left. But today he gets a really special one because ARVO represents the largest vision science and ophthalmology community around the world. And it's truly great to welcome Jose, who received the Proctor Medal 2026, and he will tell you about his work. Thank you, Jose.
Well, thank you so much to Breton. Thank you to all of you for attending, and to all my friends, and most importantly, thank you to our patients who trust us and expect us to do better and faster. So I'm very pleased and I'm very grateful to the committee who decided to give me this award, and to the people who nominated me, whom I will name. So the title may look odd, Shooting in the Dark, and I'll try to explain what it is, and also try to explain that it's not a linear process, and the Odyssey, as you know, is not a linear process. You don't even go back to the starting point, but we try to build a pathway to explore what could be done for our patient. So here was my disclosures, which I need to insist on. Some of the work was funded by companies we created, Gensight, Picture Vision, now Science Corporation, and Sparing Vision. I want to acknowledge the memory of Thierry Leveillard. We worked together for 26 years on the identification of RDCVF. It was his passion. We started that when we had this hypothesis, and I think he would be happy to be here. Maybe he will be with me on the stage to give a talk today. We tried with Denise Delcara some years ago, when we were writing a review with Breton and Jean Bennett, to lay out what could be the ways to address the needs of patients affected with retinitis pigmentosa and more generally inherited retinal degenerations. We know about the gene defects thanks to the work of many people, especially the next awardees who really paved the way to discover many genes. But when the first gene was identified by the team of Ted Raja following the work of Peter Humphreys, the rhodopsin, what was puzzling is that it could explain the dark adaptation problem, but nothing else. And if you look at the ideal therapy, it's a corrective gene therapy. We have now a few. We have one approved and hopefully more to come. But this is an early stage of the disease. And many patients, first of all, there is no gene therapy available for them, or not even in the pipeline. And second, the pathway is leading to the degeneration of cones, which is really the blinding situation. And we try to work on trying to protect the cones as a common pathway, what people call gene agnostic. Thierry hated this work. It's a gene-independent approach. Then downstream, unfortunately, many of the patients end up losing the outer segment of photoreceptors, potentially all the photoreceptors, and this is where vision restoration, optogenetic, where we worked on with Breton, prosthetic, I worked with Daniel Palanker and Serge Picot on that, and also cell therapy come into play. But when we look at that, we have to realize that the expectations are different. We are palliative sometimes versus curative. We are also trying to tackle pathways that are not the cause of the disease, so we just try to make it better for our patient. We are talking about neuroprotection versus restoration, which means that when we are assessing the efficacy of our therapies, the outcomes are very different. So I don't know what happens with my slide. Yeah. One of the issues is really all these patients with very rare disease that are away from centers, that are away from even being identified. And together with Foundation Fighting Blindness, thanks to the leadership of Jackie Duncan, we built this universal study that I presented a few days ago that is really aiming at including every possible patient across the world. There are 33 sites across the world, and hopefully there will be more, to really enroll this patient in a standardized way, in a registry where we can identify this patient. And more importantly, build upon that natural history studies whereby we can identify the markers of progression of a disease, which would help us to really define outcome measures and what could be significant for this patient. So it is supported by FFB and many companies and many family foundations, and it's work in progress. It's like a platform that all of us want to use. The work we did over the years was we tried to identify why the cones are degenerating. The roads are really the first first prime cause of a disease, and then the cones are degenerating, and then there is remodeling in the retina. And this web vision movie is summarizing what's happening in the retina, but what we try to understand is what we could do to stop the degeneration of cones. At that time, I had 5 hypotheses. One was toxicity, which is still being explored actually by Serge, and this is something which was part of our work. But the most important thing happened when we decided to try to rescue the cones. And using the technology developed by Marty Silverman, the late Marty Silverman, together with David Hicks, with Sadek Monsaid and Isabel Odo, who is in the room, we worked on trying to rescue the cones. And for that, we used the transplantation of a layer of rod photoreceptors from normal retina in degenerated RD1 mouse at a stage where the cones are still alive, but the rods have degenerated. And we found out that if you look at the transplanted area, there is rescue in this area, no connections, but you have rescue all across the retina. And then we developed a core culture model and showed that there was a diffusible protein that was responsible for that. We decided to try to identify that protein. And at that time, I remember I was giving a talk at a meeting, and one of the most prominent leaders who got all the ARVO awards was speaking after me, and he said, well, in contrast to the prior speaker, which was me, I'm not going to talk about shooting in the dark, trying to find something that nobody knows about. So I thought, does it mean to shoot in the dark? And so there are many ways to talk about it. It's coffee, I love coffee, so I thought maybe it's a nice statement, but I realized it was not that, not the nice movie either. So what is it? So if you look at the definition on using AI, you get all these definitions, and for many people that get the summary statement from the NEI or NIH, this looks very familiar, like an action with an unpredictable result, a guess without any information or certainty. So this is the type of thing we get when we try to do something, which is kind of discouraging. But I'm Jewish, as you know, and I tend to believe that we should never ask our way to people who know it, because we might fail to get lost, and getting lost is sometimes the best way to get somewhere. And so we embarked with Thierry on a very systematic approach, trying to identify this factor, and we came up with this idea of using expression cloning, which is expressing all the genes in the retina in a library, taking the supernatant, and using a model that had been developed by the late Ruben Adler at Hopkins, using cheek embryo cone photoreceptors, screening by 100 and then 10 and 1, until out of 200,000 clones, we were able to identify the factor we are looking for. When we tested it, once we sequenced it, at that time this was the years where the genome was being sequenced, it turned out it was a new sequence, totally unknown at that time, which was put in the files. And this gene is actually encoding for 2 types of protein. The one we identified was the short form that is really expressed after intron retention. It's only one part of a gene that is expressed. It's a short form of a disease, and this is a trophic factor that is which is really the interaction between the rods and the cones. But there was also a longer sequence, which is actually cell autonomous, and it turned out it was really very similar to a thyroidoxine, so which led us to a trophic approach and the antioxidant approach. We tested that in the RD1 mouse, and it was working using the protein. We tested that in the rhodopsin mutant rat, and we found out that there was a rescue of cone photoreceptors, but there was even a better functional rescue. We didn't know why. The reviewers asked us to explain. We found out that the outer segments were longer in the treated retinas versus the untreated retina. At that time, we didn't know why this was the case. Then Thierry, together with a team, and especially Najat, who is now with us in the room and now with me in Pittsburgh, embarked in trying to identify the receptor of RDCVF because it is a cell-mediated exchange. And eventually, after almost 8 years of work, this receptor was identified. It was an orphan receptor, Vasicin-1. And looking at the machinery and what happened in this beautiful paper, what was identified is that Vasicin-1 is activating the entry of glucose into the cell, activating GLUT1, the transporter, but targeting a very specific pathway. Yesterday, John Nash was talking about all the metabolism of glucose in photoreceptors. There is a specific pathway that at that time was not ignored but neglected, called aerobic glycolysis. It's making carbohydrates that are used for the buildup of the outer segment of photoreceptors, and this does explain why we had this longer outer segment after the therapy. It also provides lactate that goes to the RPE and gets to the choroid. And this aerobic glycolysis had been identified by Otto Warburg in the 20s in Germany. He's one of the few Jews that Hitler didn't want to kill because he thought he would develop a treatment for cancer, because Otto Warburg believed that glucose metabolism was important for cancer cells to develop, and there is a book about it. But he also stated that it was never happening in a normal cell, but he had one artifact, which was the retina. He thought this was an artifact. It turns out it's not an artifact, and there's a review by Krebs, a Nobel laureate, that really said this is an artifact. It's not. Retina is using glucose to build outer segments. So Connie Sepko commented on our paper, and she built this cartoon, which I think is very nice, explaining the idea that is behind the treating hypothesis we have, expressing RDCVF to really rescue the cones, binding to remaining receptors on the cone to treat the patient. But remember, there was another form of a gene, which is a thyroidoxine. So we looked also at the oxidative stress, and the knockout mouse of RDCVF, NXNL1 knockout, is actually extremely sensitive to oxidative stress. We could rescue it by gene therapy expressing the long form of RDCVF, which tells us that you'd probably need the two forms of the gene. And indeed, we launched a collaboration with John Flannery and two of his students at that time, postdoc and student, Denise Dalcara and Leah Byrne, who eventually joined me later on, one in Paris, one in Pittsburgh, worked with John on that. And this led to demonstrating in this JCI paper that the two forms of a gene are very important for functional rescue. And if you look at the untreated, the PBS treated versus the RDCVF treated, there is a clear difference in the density of cones, but more importantly, the ERG is rescued, and there is functional testing also in this paper using optokinetic. We then built, with Thierry, a combination of the two genes into one construct. And again, we could show that in contrast to one only, there was a better rescue with a combination of the two. We created a company, Sparing Vision, that developed the construct, but is really putting into one construct the short form under a ubiquitous promoter to express it in the RP cells, and the short form and the longer form, which is driven by a cone photoreceptor specific promoter to express it in cone photoreceptors. And we showed, using optokinetic, that there was a significant rescue, not total rescue, but significant rescue. This led to propose a clinical trial, and this is a cartoon by Thierry that we put in a review we wrote many, many years ago. In parallel, other groups are working on the antioxidant, which is familiar to us, beta-camptocarone, Acuity, for example, and also Connie Sepko. Connie has been working on glucose metabolism, following an independent pathway that is converging with what we demonstrated, and also inflammation. And there are many others using transcription factors also, so these gene-independent approaches are still in the pipeline. So with Sparing Vision, we launched 3 types of studies, and Isabelle is leading many of those in Paris. One is a natural history study looking at retrospective data from the patient we have in the huge database of patients we are following in Paris. And then a prospective study called PhenoROD, looking at patients affected with rhodopsin mutation and phosphodiesterase mutation. We selected 2 mutations, two genes, actually not mutations, 2 variants in genes. And then Prodigy, which is the first study to really do a safety study, a dose escalation, and an extension cohort, which is now fully enrolled only a few months ago. So the data are being analyzed using different types of models, statistical models, machine learning. The idea is to identify fast progressors based on this natural history study so that the data from the patient in the study could be analyzed. At this stage, we have safety data from the first 3 cohorts, and we start to have some early signals, positive, but too early to draw any conclusion, but at least it's good to have positive initial trends. Beyond that, I mentioned that rescuing the cones would be wonderful, but there's also a stage of the disease where the cones have lost the outer segment. They are no longer responding to light, and Boton Roska in his lab had a postdoc. We came up with this idea of trying to reactivate these cones. And Boton and I discussed about that, but it was a great idea, a crazy idea, but a great idea. And the idea was to use that. Interestingly, many people thought that dormant cones don't exist. People who are blind don't have any cones left. Matt Lavelle sent us 12 papers demonstrating that in many published studies, there were dormant cones in advanced models that nobody really looked at. So the idea is to use optogenetics. It's using proteins that exist in algae and exist in rudimentary organisms that are responding to light and are able to trigger an electrical response throughout the membrane. Sorry, it's going back. Yeah, and so it's replacing the full phototransduction, but it's not really doing everything in the phototransduction because there is no light adaptation. But with one protein, you can really provide a lot. And so the idea was to express these proteins in remaining cells in the retina, in that case, the dormant cones. So this was the first strategy we attempted, targeting the cones. Actually, at the Vision Institute and with collaborators, we targeted every cell type in the retina. So this paper came out 16 years ago. We are very enthusiastic about this idea that you could rescue the cones, express a heterodopsin, which is a pump, hyperpolarizing in response to light, and get a visual behavior and recording from the ganglion cells. Volker is in the audience, and he was part of that work with David Belia, who unfortunately passed away. And this was led by Botton, and in Paris with Serge Picot, we tested that in ex vivo retina from donors. But we checked in our database of patients at the Vision Institute with Michel Pack and with Isabelle, and it turns out that we had around 20% of our patients who were legally blind, or totally blind actually, but still had dormant cones, so a potential target. Unfortunately, when we tried to get that into a clinic, we faced a lot of issues with expression of a protein in the primate, not in the mouse, and we decided to explore alternative avenues. One of these was to go to ganglion cells. The rationale for that was built many years ago, where each of us were looking at the OCT, and there is a beautiful paper by Sam Jacobson and Artsy Desian showing that you have remaining ganglion cells even at very advanced stages of a disease, which is what is in orange on the left-hand side. And the idea was to target the remaining ganglion cells using this time a channel rhodopsin, which is a depolarizing protein. So we were not the first to think about that. Actually, Pan in Detroit thought about it and published a paper where he showed actually that you could get some electrical responses in the mouse, in neuron. And unfortunately, the amount of light you need for that was immense, which is probably too much for a patient, although this led to creating a company and start a clinical trial whose results I never heard of. We tried to optimize that, and Ernst Baumberg, who was one of the inventors of optogenetics, had built CATCH, which is a protein that is optimized for light responses. And with Denise and Serge, we showed that you could get very good responses at the retinal level, at the cortical level. But the amount of light was still too much. So we were looking for an amount of light which would not be like staring at the sun in the desert or on the snow. And we were looking for a protein that might be shifted into the red, the longer wavelength, because the energy might be less. And at that time, Roger Chen in San Diego developed RICHAR, which was tested by Jens Stubel on bipolar cells. And in parallel, Ed Boyden at MIT had developed several proteins, including Crimson R, that was shifted into the red. And Ed Boyden contacted us and offered us to try it. So we tried it, it looked very good. And this is what we decided to do because the safety range was much better. Then Denise was screened a lot of vectors, and I'm not going to bore you with that, but eventually the 7M8 variant that had been identified at Berkeley was the best one to target the ganglion cells. So then this was tested as a construct ex vivo in treated retina for primates, demonstrating that you could actually decode the information at the retina level on multi-electrode arrays and build a mathematical model, a Bayesian model, a decoder, and predicting the level of vision. In this very artificial model, the visual acuity you could expect would be actually in the range of 20 to 100, which is not what we are seeing in the clinic because this is a very artificial model. But this led us to build a clinical trial with GenSight, a company we founded, and the idea was to use a combination therapy of a gene therapy to deliver to ganglion cells the construct, use goggles that were developed by our team to stimulate actually the retina in the wavelength corresponding, and the trial was started. It just started at the initiation of COVID. Most patients were lost to follow-up, but one patient living next to the hospital started to report that he could see the crosswalks in the street using the goggles. So we brought him back, and at Street Lab, the team looked at what he was able to do, and he was able to see object, to grab object. We decided not to say anything about it, to test again, to work on it, and we were suggested to use multi-electrode encephalography to monitor what was happening at the cortical level. So eventually, other patients were demonstrated the same thing. This is another patient in the cohort who was able to really see quite precisely, and we decided to pair that with objective assessment. So here you see the cortical activation in the patient in the trial, and you see that only the occipital area is activated with a desynchronization of waves that corresponds to vision, and it's far above chances. So then we decided to standardize for all the patients in the study the assessment based on what we were learning from the patient, so that we could have a very specific systematic approach. What objects are being grabbed, the size, the contrast, and then also looking at the screen. And the paper is close to be published, but this is about the cohort of patients. Most of them had a very significant improvement, more than half of them. A few didn't have a significant improvement, and one didn't get much better. And importantly, we also did FST in this patient, and across the 9 patients tested, 7 of them had an improvement in the FST, very significant, even without the goggles. So this is, of course, not enough to daily life activities, but this is very promising, and this is part of the adventure of optogenetic. Now we have many competitors, many companies are working on them. Some are still alive, some are dead, some are very promising. There are new companies coming into that, Restore Vision, Sparing Vision, I will talk about that, and Right Gaze, for example, and many others. Going back to how we can do better, we decided to improve the resolution, and we have a very promising project ongoing in Pittsburgh with Morgan Fedorchak, Leah Byrne, and with Valentina Emiliani in Paris, trying to optimize the optogenetics. The goal this time is to address exactly the part of the retina we want to target, so to use a gel to really paint the part of the retina that is going to be transduced. Also to use specific subtypes of vectors that are going to target specific pathways instead of all cell types in ganglion cells, for example, and using holographic, digital holography and 1-photon and biphoton microscopy to stimulate individual cells with eye trackers. All of this has been funded by NR01, the DoD, and the PTINC, which is a Pittsburgh support, and this is showing very promising results that we are trying also to bring into the clinic, but this is going to be a significant journey. In parallel, Denise didn't give up on cone optogenetics, and she got the idea of using a G-coupled protein that is existing across the body, but not specifically in the retina, called GERK, which is a rectifying, inward rectifying potassium channel, and to use that in the dormant cones, and to try to see if you could reactivate the dormant cones using this approach. And on the left, you see the result on the ERG and the result on the optokinetic. There is a significant improvement in visual function in the mouse, in the RD mouse, RD1 mouse. Of course, the cones continue to degenerate, but if you look at the lifespan of a mouse, it's a very large duration in terms of effect. This also led to a clinical trial with Sparing Vision that acquired actually this program, that is now in the clinic. 2 patients have been enrolled and treated in the first cohort. It's a dose escalation study. And again, we have early signals that there is some, we took totally non-light perception patients, which are very few, actually very difficult to find. And there are promising data from the 2 first patients in Ghent and in Dublin, but again, too early to conclude anything at this stage. So I've tried to do many things, and many things have failed. So at this type of lecture, you don't talk about failures, but I can talk about overshots in the dark. Now people talk about moonshots. Looks a bit more positive than shooting in the dark. And among these moonshots, some are trying to improve outcome measures. There's a huge endeavor, many centers, many companies, but also FFB with a ready initiative trying to really define the best outcome measures. Some of these are using structural markers or functional markers, and this concept of functional vision that was behind the approval of Luxturna is a very important one. So we built, for example, this platform in Paris, the Street Lab, and developed this virtual reality system to monitor the impact of vision loss in daily activities, trying to give a voice and a representation to what patients are describing based on the feedback of 1,500 patients that really describe what they were experiencing. And this is now being used across many sites for trials and for natural history studies. But other moonshots are prostheses and eye transplantation. So of course, you need to do that depending on what you know on the status of photoreceptors, and very important efforts are being conducted to assess the status of photoreceptors and the rest of the retina in our patients. So I won't speak a lot about artificial retina because you'll have opportunities to hear from Daniel the beautiful work that we were able to continue together based on his invention of a Prima implant. So the Prima implant is a photodiode array that is implanted under the retina at the location of photoreceptors with 400 electrodes, each of them independent with their own amplification and their own activation. And it was decided to try it in patients with advanced age-related age-related macular degeneration, the atrophic form, because these patients don't have any central vision, they still have peripheral vision. So this has been done throughout many clinical trials following technology development with Fiction Vision and then Science Corporation. So there were 3 trials that were conducted. The first one was conducted in my former department at the Rothschild Foundation in Paris by Yannick Lemaire, implanting 5 patients. This has been published already. And we could show that in several of these patients, there was a very significant improvement in central visual acuity, especially using zooming and improvement of the images. But there was also a preservation of peripheral vision, which means that this patient could combine their peripheral vision with their central vision. We did a second study with Jose Martel in Pittsburgh, confirming the first one. And then this led to a large clinical trial across Europe in the 13 sites, 5 countries, and 38 patients were implanted at end-stage age-related macular degeneration. This was led by Frank Holz, published a few months ago. And I'm giving a talk on the subgroup analysis on Wednesday, and there will be many presentations on that upon ARVO. But just to give you an idea of the effect, the average improvement in patient was 25 letters. One of the patients, exceptional, a French patient, got 59 letters, which is 12 lines, but the average was this range, so far above the expected outcome measure, which were 2 to 3 lines of improvement. And this is only with the implant and nothing else. And this is one example of one of the patients able to draw, actually to do crosswords using the implant, but we have many movies showing examples of daily life activities. It's working in around 80% of the patients in this trial, and this is leading to obviously pathways towards approval and potentially others. Another moonshot, but this time this is the name that was given to it, is the eye transplantation program. So you'll see there are many presentations across the meeting. We are one of the consortia that is funded by ARPA-H, the new agency that is supporting this type of crazy effort. And they say the future of health isn't waiting, and never are we. So that's why we should never wait. We should work. And this is led by Jeff Goldberg with Vijay Gorantla, and John Ash, who is helping me to do that. So we are leading it together, trying to address the multiple challenges, finding the best donor, keeping the retina alive, developing the surgery, trying to make sure that the surgery is done fast enough so that you are really able to get that into the patient on time, mastering the immune response, and the most important and the most daunting task, but we have amazing data from that, is regenerating the optic nerve. I think there's a talk by Taka from our department on Thursday on that, but there are many talks about this type of data. This is the type of crazy project that initially you think that we should never do, but eventually it yields a lot of information, a lot of things that can be useful to our patients potentially. So in closing, I just want to say it's an Odyssey. So there is a beautiful poem, a Greek poem that many of you, I guess, know about traveling and the journey. So I'm sorry if I have to read it for people who have low vision. As you set out for Ithaca, hope your road is a long road, full of adventure, full of discovery. May there be many summer mornings when, with that pleasure, what joy, you enter harbors you are seeing for the first time. Keep Ithaca always in your mind, but don't hurry the journey at all. Better if it lasts for years, so you are old by the time you reach the island. So it is happening to me, I'm old now. You have understood by then what these Ithacas mean. So there's only one thing I disagree with this poem. It's good to take time, but our patients are not willing us to take time. So it's a long journey, but we have to do faster. To do that, in contrast to Lise, who lost most of his friends and in the Odyssey, I've been fortunate to work with many people. A few of them, like Thierry, have disappeared. And I put just a few faces on this slide. I want to acknowledge especially my mentors, Dan Albert and John Dowling, who is in the audience today. I'm very moved to have him in the audience. People who helped me, people who work with me in Pittsburgh, people who work with me in Paris, in Strasbourg, and many places, many friends. And I want to thank especially my friends from Jerusalem, Itai Howers, who nominated me for this award, together with Don Zak, with Botan Roska, and Serge Picot. But I should put on this slide too many faces. So this is the list of names I just tried to put together, people I worked with over the years. I benefited from them. But I certainly want to insist on the most important partner we have always had in life, which is our patients. Thank you very much.